Okay, so in this video we are going to discuss about glycolysis. Now this video will come under two parts. In part 1 we will discuss the basic things, that is the steps, sequence, enzymes involved, the definition of glycolysis, everything. And in the second part we will discuss about other things such as the inhibitors of glycolysis, the regulatory enzymes, substrate level phosphorylation, oxidative phosphorylation, all this. That is the questions that are frequently asked related to glycolysis. Now the meaning of the word glycolysis. So as you can see, glycolysis, so it will break into glyco part and lysis part. So glyco is coming from glucose or any other sugar materials, and lysis means breakdown. Now sugar materials is mostly glucose only, because glucose is the simplest form of carbohydrates, and any form of carbohydrate that we take is converted into glucose only. So in a simple term we can say that breakdown of glucose is called glycolysis. Now it has got some other names. One name is Emden Meyerhof Parnas pathway, or we can also say EMP pathway. So this is other name, this is a breakdown, that is the origin of word. Next I will just talk about the location of glycolysis. It occurs in the cytoplasm of cells. Cytoplasm, it is also said as cytosol, so don't get confused. So this is also a frequently asked question, that where does glycolysis occur? Cytoplasm of cells. Now of which cells? It can be anything, that is your kidney, your retina, lens, kidney cortex, medulla, red muscle mass, etc., etc. So it occurs in the cytoplasm or cytosols of these organs. Some more you write over here: white striated muscles, erythrocytes, skin, brain. Just remember some of it. Next we come to the definition. Now in any definition of biochemistry or anything that you give, you have to keep in mind that in the definition, the starting material, the ending material, the location, and any condition, that is if it is required, that is in the presence of oxygen or in the absence of oxygen, this thing you have to give. So this is the basic rule of any definition that you provide in biochemistry. So the definition is: glycolysis is a breakdown of glucose, that is hexose sugar, in the cytoplasm of cells. These examples you can give, in the absence of oxygen. So basically anaerobic process, but it can also takes place in the presence of oxygen. If oxygen is present, then the end product of the total glycolytic pathway is pyruvate, and if it is taking place in the absence of oxygen, then the end product is lactic acid. So we will come to know about that later on. So in this part we discuss the other name, the breakdown of this, location and definition. Now this total process of glycolysis is divided into 10 steps. First 5 steps and the last 5 steps. The first 5 steps is known as the preparatory phase or the investment phase, and the last 5 step is known as the payoff phase. We'll come to know about that. Now we'll talk about the preparatory phase or the investment phase. The first 5 steps is the preparatory phase. So here glucose is the starting material. The structure of glucose you have to learn. Glucose is converted to glucose 6-phosphate by these enzymes, hexokinase or glucokinase. Option. Both are not required, only one is required. Now, what is the difference between hexokinase and glucokinase? Hexokinase will act only on all hexose sugars, but glucokinase will act only on glucose. This is the difference. More differences we will come to know in the enzyme chapter. Now glucose is converted to glucose 6-phosphate by this enzyme. It also requires another coenzyme, which is ATP, adenosine triphosphate, and it also requires a cofactor. A cofactor, coenzyme, enzyme, everything will be learned in the enzyme chapter. Just you know these things, that cofactor and coenzymes are those molecules or ions that help the enzyme to work for its catalytic activity. So cofactor is either magnesium ions or manganese ions. Now this is the structure of ATP. Adenosine part, this is the ribose part, and this is the 3-phosphate groups. Now what is happening over here? How this glucose is changing to glucose 6-phosphate? You have to know it properly. So here glucose is converted to glucose 6-phosphate. That means in the 6th position phosphate is coming. Therefore we know that this is 1, 2, 3, 4, 5, this is 6. So here only the changes will occur basically. This pen is very light. Okay, we'll use this pen. So in this position the changes will occur. What type of changes? This H, as you can see here, this will break this bond. H will be over here, and this part, that is, see, amader একটা phosphate chai, we require only one phosphate to make glucose to glucose 6-phosphate. Therefore this part will come and attaches over here, and H, this part will remain, so one phosphate molecule less, so it is converted to ATP. So every structure is same, only if you put here H, the structure will be of ADP, adenosine diphosphate. So this is done. Next is, as you can oversee, therefore one ATP molecule is converted to ADP. We will consider all these things in the energetics part. Just only remember now that during the conversion of glucose to glucose 6-phosphate, one molecule of ATP is utilized and ADP is generated. Next, in the second step, glucose 6-phosphate is converted to fructose 6-phosphate. Here the enzyme that is required is, now the enzyme is phosphoexose isomerase. Isomerase enzyme, glucose and fructose are isomers, so it will change. Nothing is there, only as you can see, glucose 6-phosphate will change to fructose 6-phosphate. Here is the 6th position, phosphate is there. Next, fructose 6-phosphate will be converted to fructose 1,6 bisphosphate. Now, in many books here, in the place of bisphosphate, you can also see diphosphate. But what is the difference between the two? Bisphosphate means in a molecule, 2 phosphate molecules are present, both in this case and this case. 2 phosphate molecules are present. Now, when the phosphate molecules will be placed one after the other, that is attached to the same carbon, as in this case, see, same, one by one they are attached. So then you can use diphosphate, triphosphate. But whenever it will be attached to different, one to this and one to this, we use bisphosphate. It's not wrong, but it is more specific. It is more correct, right? Next, the same thing. Here also, ATP will be converted to ADP. Same cofactor will be used. Whenever ATP to ADP is there, Mg2 plus Mn2 plus will be used. In the case of the reverse also, this thing will occur. That means from ADP to ATP. Next, fructose 1,6 bisphosphate. This is the ring structure, but it will be more clear if we see this thing. The straight chain. What will happen? This H will shift to here. Therefore, it will become a double bond and this will be cleaved. And this particular thing will be joined to form another molecule. So, 2 molecules will be formed. Dihydroxyacetone phosphate or glyceraldehyde 3-phosphate. This part will become DHAP, that is dihydroxyacetone phosphate, and this part will become glyceraldehyde 3-phosphate. So this is the first 5 steps. It will also interconvert, that is included in this part only. All DHAP molecules will be converted to GAP molecules, right, by the enzyme phosphotriose isomerase. Now, why it occurs? Because the next enzyme that will be there, I will talk in the next part, that will only act upon GAP and not on DHAP. So, therefore, this conversion is necessary. But here, one thing you have to note down, that from this single molecule, 2 molecules are generated, and this is also converted to this. So, from here, whatever you will do, you will add 2 in front of that, because now there are 2 molecules of this thing. Again, a small part, energetics. It is linked to these things only, but ekhane amra just we have seen that in the first step, 1 ATP is utilized, that is minus, and in the third step, another ATP is utilized. Keep in mind these things. No other ATP are included in this case. We will concentrate on the payoff phase, that is the last 5 steps. Now, till this thing we have learned that from dihydroxyacetone phosphate glyceraldehyde 3-phosphate is formed. Mane glyceraldehyde 3-phosphate is formed, but also DHAP is also converted into glyceraldehyde 3-phosphate. Now I have already said that you have to add 2. Now this particular thing is oxidized to an intermediate compound. This is oxygen, nascent oxygen, 1 O will be added. So this is the oxidized intermediate product. This is 2-pi, means this is the formula. This is inorganic phosphate. 2-pi will be there. This is also 2. Here also 2. Everything will be 2. You can either give here 2 to 2, or you can write into 2. Now 2-pi, so 1 H from here and 1 OH, that is 1 molecule of water will go out, and will this entire thing will be attached over here. So this O is there, H is not present. Instead of this particular thing is present, and the H that was present here, it has formed H2O. So it will form 1,3 bis-phosphoglyceric acid. The enzyme that is used over here, it is glyceraldehyde 3-phosphate dehydrogenase. So this will be formed. Now here NAD plus is converted to NADH plus H plus. So this is the coenzyme that is required over here beside the enzyme. The full form of NAD plus, nicotinamide it is nicotinamide adenine dinucleotide. Now whenever it is NADH, that is the reduced form, that is H is added. So addition of H or removal of oxygen is reduction. Now it will be formed. I will come later on the energetics part. Here also 2 will be there. Next হচ্ছে 1,3 bis-phosphoglyceric acid will be converted to 3-phosphoglyceric acid. Here ADP will be converted to ATP. Right? How? If you refer to the structure of ATP, this total part, that is from the first position, because 3-phosphoglyceric acid is there, so from the one position phosphate has to be removed. So it will be removed and OH will come over here, and ADP, as ADP is there, so one phosphate is going to ADP, so it will definitely form ATP. The enzyme involved is phosphoglyceric acid kinase. I should change the color. Okay. 3-phosphoglyceric acid is formed. Now 2-phosphoglyceric acid will form, and here also an isomerase enzyme will be involved. Phosphoglyceromutase is the enzyme. It will change to 2-phosphoglyceric acid. Next, this OH and this H will go out. H2O will go out and it will form this. The name is PEPA, that is phosphoenol pyruvic acid. This will be reacted upon by pyruvate kinase. Here also ADP to ATP, and Mg2 plus Mn2 plus is constant over here. Here also Mg2 plus Mn2 plus. Now here it will be converted to, this is also pyruvic acid, but this is unstable form. Tautomerism and hydrogen shifting will occur. This H will shift over here and this double bond will be shifted over here. This is the stable and it is the keto form. Now, one more thing you have to learn. Here, NADH plus to H plus will be converted to NAD plus. 2, 2, 2, 2, 2, 2. Everything will be 2. Even the water will be also 2. ATP, ADP, everything will be 2. Everything, okay? So what will happen over here is that in anaerobic condition, when there is no oxygen. Now here we can see NADH plus H plus is produced. So there is no NAD plus now. This particular thing has to be oxidized. That means reoxidized in the electron transport chain, about which you will learn later on. But there is no oxygen over here. Then also glycolysis process has to be performed. Otherwise how one can get energy? Then this particular thing will occur where this is getting converted to NAD plus, so that this process at least can go on in absence of oxygen. That is, pyruvic acid is converted to lactic acid in absence of oxygen. If there is presence of oxygen, then this step has got no requirement. Then you can skip this step. Pyruvic acid is formed, it will enter the Krebs cycle. We will learn about Krebs cycle later on. Just briefly revise this thing. This is the preparatory phase and this was the payoff phase. Now energetics part we have already done that in the first step, that is when glucose is converted to glucose 6-phosphate, 1 ATP is utilized, and in the third step where fructose 6-phosphate is converted to fructose 1,6 bis-phosphate, 1 ATP is utilized. Next, again another thing is over here. This I am coming later on. Here as you can see that when 1,3 bis-phosphoglyceric acid is converted to 3-phosphoglyceric acid, ADP to ATP conversion is taking place. Now as 2 is there, so that is 2 ADP, 2 ATP. Okay, so let us write this over here. Now in the seventh step, that is where 1,3 bis-phosphoglyceric acid is converted to 3-phosphoglyceric acid. ADP to ATP conversion is taking place, and as you can see over here 2 ADP to 2 ATP, that is plus 2, because here ATP is generated, in this ATP were utilized. In the ninth step, that is this one, when phosphoenol pyruvic acid is converted to pyruvic acid, again 2 ATP from 2 ADP 2 ATP is produced. So here see plus 2. Now in the sixth step, that is where NAD plus is converted to NADH plus H plus, we have to know this thing that when this thing is going to the electron transport chain from one molecule of NADH plus H plus 2.5 ATP are generated. So here 2 NADH plus 2 H plus is from, so 2.5 into 2, that is 5 ATP. Now we come to the energetics under two conditions, in the presence of oxygen or in the absence of oxygen. So now I will discuss about the energetics of glycolysis. So here we will discuss about the ATP generation and utilization. Okay. So as you can see over here, we have learned that when glucose is converted to glucose 6-phosphate, 1 ATP was utilized. That is, from ATP to ADP was formed. That means 1 ATP was utilized. So this is done. In the second part, fructose 6-phosphate to fructose 1,6 bisphosphate, again from ATP, ADP was produced. So here also 1 ATP was utilized. Next, in the glyceraldehyde 3-phosphate to 1,3 bisphosphoglyceric acid, in that step, this thing was there. What was the thing? NAD plus is converted to NADH plus H plus, right? So as I already said that 2 will be there, so 2 NADH plus 2 H plus. Now this when will go to the electron transport chain, that is ETC, obviously O2 should be present, then only it will go to the ETC to get reoxidized. Then from 1 NADH plus H plus, 2.5 ATP will be produced. So as there are 2 molecules of NADH plus 2H plus, therefore 2.5 into 2, total 5 ATP will be produced. This particular thing will occur only when there is oxygen, so that this thing can go to the electron transport chain to get reoxidized to release this ATP. Next, 1,3 bisphosphoglyceric acid to 3-phosphoglyceric acid. I already said here 2 molecules are there. So 2 ATP, because here 2 ADP to 2 ATP was produced, therefore 2 ATP total. Next, phosphanol pyruvic acid to pyruvic acid, also 2 ATP, because here also 2 ADP to 2 ATP, this conversion has taken place. So now the energetics, that is the total number of ATP generated, can be categorized under two conditions: aerobic condition whenever there is oxygen, and anaerobic condition when there is no oxygen, right? So in aerobic condition, the energetics will be as follows. So now just tell me, whenever there will be oxygen present, everything will occur. This will also occur. These steps require no oxygen, but this particular thing requires oxygen to be present. Therefore everything will occur in aerobic conditions, and in anaerobic condition only this part will not occur, because this part will occur only when there is presence of oxygen, so that this particular thing can go to the electron transport chain to release or generate this ATP. So we can simply write in the aerobic phase that this will be minus, because here ATP were utilized. So generated is 2 plus 2, that is 4. This is also in the generated part. So we can say 2 plus 2, that is 2 plus 2 plus 5 minus 1 plus 1, that is 2. So it is 4, 9. 9 minus 2, that is 7. So this is the net production. And if you say about the gross, so this is the gross production, that is without minus. So this is the gross production. So you can be asked, what is the gross production, what is the net production? Then don't get confused. Gross means without the subtraction, the total produce 5 plus 2 plus 2, 9. But whenever it is net, that means this has to be subtracted. Okay. Next, in anaerobic condition, everything will occur only except this 5. So it is simply 2 plus 2 minus 2. Only 2 ATP. So you can write over here that in aerobic condition, net production is 7 ATP. And in anaerobic condition, net production is 2 ATP. So this is actually the end of the first part of the glycolysis. In the next part, we will be discussing about the steps where oxidative phosphorylation is there, substrate level phosphorylation, as I already said, all these things. So if you like this video, do like, subscribe my channel, and if you want anything extra, you can comment in the comment section below. Thank you.